Hello, this is a presentation of W2S microscopy data with joint denoising and super resolution for wide field to SIM mapping, carried out at School of Computer and Communication Sciences, EPFL, Switzerland. Real super resolution is always subject to noise, although of varying intensity. There was, however, no benchmark for studying the joint task of denoising and super-resolving a noisy image with non-negligible noise. Such an application is very important in live microscopy, where excessive illumination is detrimental to the captured subject. Thus, the microscopy images usually suffer from very high noise and limited resolution due to the friction of the capture devices. To study joint denoising and super resolution on microscopy data, we collect the wide field to SIM W2S dataset containing low-resolution images, each with varying noise levels and the corresponding high-resolution images. This allows us to benchmark many candidate solutions and to develop a joint denoising and super resolution method. We collect the W2S dataset using wide-field microscopy. Images were acquired with a high-quality commercial fluorescence microscope and with human cells. For the low-resolution images, we capture the cells with a short exposure of 5 milliseconds, thus each raw low-resolution image contains a strong noise component. 400 shots are taken for each field of view. As the noise is zero mean, we can average a varying number of samples to generate low-resolution images with different noise levels. Thus, we perform averaging on the raw image data to obtain low-resolution images with different noise levels. The denoised low-resolution images are evaluated by comparing to the average of all of the 400 raw captures. For the high-resolution images, we use a microscopy technique, structured illumination microscopy, or SIM for short. SIM is a technique used in microscopy imaging that allows samples to be captured with a higher resolution than the one imposed by the physical limits of the imaging system. We use SIM to extend the resolution of standard wide-field microscopy images. This allows us to obtain aligned low-resolution and high-resolution image pairs, thus enabling the benchmarking of both denoising and super-resolution methods under real settings. Here we show an example from the dataset, where we have low-resolution images with different noise levels. The ground truth low-resolution images is estimated by averaging the 400 raw images. And we also show the high-resolution SIM result. W2S includes 120 different field of views. Each field of view is captured in 3 channels, corresponding to the wavelengths 488 nanometer, 561 nanometer, and 640 nanometer. As the texture of the cells is different and independent across different channels, the different channels can be considered as different images, thus resulting in 360 views. We obtain low-resolution images with different noise levels by averaging a different number of images of the same field of view and the same channel. We average 1, 2, 4, 8, and 16 images to obtain low-resolution images with 5 different noise levels. As the device is well stabilized throughout the capturing process, the low-resolution and high-resolution images are perfectly aligned. To quantitatively evaluate the difficulty of recovering the high-resolution image from the noisy low-resolution observation in W2S, we also analyzed the degradation model relating the low-resolution observations to their corresponding high-resolution images. The statistics of noise estimation and kernel estimation of W2S and other real-world datasets are shown in the figures. W2S contains a comparably high noise level and a wide kernel, which is shown in green color, indicating that the high-resolution images of W2S are challenging to recover from the noisy low-resolution observation comparing to the other datasets. We benchmarked the sequential application of state-of-the-art denoising and super-resolution algorithms on W2S. For denoising, we evaluate different approaches from both classical methods and deep learning methods. For the super-resolution methods, we benchmark on 6 representative super-resolution networks, including both distortion-based networks and perceptual quality-oriented networks. Furthermore, to test the effectiveness of the joint optimization of denoising and super-resolution, we also trained 3 super-resolution networks on the joint denoising and super-resolution task, including RDN, ESR-GAN, and an RDB network trained using a novel texture loss. Our benchmark results show that state-of-the-art super-resolution networks perform very poorly on noisy inputs. Both quantitative and qualitative results show that super-resolution networks are very sensitive to noise, and the sequential application of denoising and super-resolution algorithms is suboptimal and suffers from the accumulation of errors from both stages. One interesting observation is that applying the best denoiser in terms of reconstruction error followed by the best super-resolution method does not necessarily yield the best final result. We also observe from the experimental results that the networks benefit from joint optimization for denoising and super-resolution, which yields the best results on our benchmark. In summary, W2S contains raw images for training real denoising, real super-resolution, and the combined joint denoising and super-resolution on cell data. We benchmark and analyze different strategies on a variety of methods, including our own, and W2S enables training-based methods for obtaining structured illumination images from wide field captures. To know more about W2S, check out our paper and code. They are publicly available.